Thank you for joining today. This workshop series is really focused on understanding analysis with an emphasis on automation. The aim is really to equip you with tools to allow you to save time and move away from potentially biased manual approaches, allowing you to perform rigorous research efficiently. And we're focused on using open source tools because anything you develop with them can be easily shared, reused, and validated by the scientific community. But it also allows you to transfer your research methods. So when you move to a different university or a different institute with different resources, you can still perform the same research that you were doing in your current role. But today, I wanted to sort of expand on this and help you learn how to use imaging and analysis in the same pipeline. And we're going to do that using a commercial software package called NIS-Elements, which is the software that Nikon uses to control their microscopes. And we have this installed on our spinning disk and slide scanner and another sort of new high-speed upright system. And so for the workshop today, we'll be using the W1 spinning disc confocal. And so hopefully some of you who are joining today are already using this system. And it's really a perfect system for this because it has very high sensitivity SCMOS cameras. So we have a large field of view, we've got a lot of sensitivity, and we can image optical sections very quickly in tissue and in cells. And so when you start doing things like that, you can start to think about your research in a different way. Instead of sitting at the microscope and manually collecting data, you can start to think about automation and then thinking about what it is you actually want to get from that data, and then start to develop an imaging and analysis pipeline that really targets getting that information efficiently. So when you first start to use elements, one of the tools we teach is this ND acquisitions tool. And this is a sort of really robust and reliable tool for doing automated experiments. It can be very effective when you start to sort of do Z-stacks and multi-point imaging and large imaging all together to sort of image sections or regions of interest in an automated way. But it is still a little bit limited, because a good example would be when you're trying to image tissue sections from an organ like the brain, where different sections can have different sizes. If you use something like ND acquisition, you'll have to use the same kind of large image window to capture all of those sections, and that slows you down and also creates a lot of unnecessary data. And you may also want more control. So maybe you're trying to find rare events in a tissue section. So maybe you need to look at the tissue section and then find where these rare events are. And you want to do that without having to be at the microscope. So you could set something up where you first detect the tissue section, then run another step where you find these cells or the injection site or the expression site, and then go into higher magnification and capture those regions. And all of that can happen autonomously. And the other thing is you may just want to perform the analysis at the same time, so that when you leave the microscope, you have the results in hand. And so the way that you can start to think about these projects is using a different tool called jobs. And you may be already somewhat familiar with these from the jobs that we built when we purchased the system. And a good example of that is the jobs that we have for automatically scanning slides so that you can then focus on tissue sections. But the idea is just today to introduce you to how you can use these jobs yourself and start to think about how you might want to apply them to your research. And of course, we'll go through some simple examples today just to kind of highlight what's possible and then get you more interested. And of course, we can help you down the line in terms of building more complicated ones. And the sort of second step to this is, so jobs is about automating what's happening at the microscope in concert with analysis. And the way that you can do analysis really powerfully in elements is using a tool called general analysis. And more specifically, this general analysis 3 is a new approach that has been recently launched. And if you've been following along in this series where we've learned how to use macros and Python scripts, this is a much easier learning curve because it's very graphical. And so all of the different tools and analysis modules can be laid out in a sort of two-dimensional map, and you can connect these components together. And so very quickly, you can end up with a very complex and powerful pipeline that you can then start to apply to your data, while you're imaging, but also after the fact. If you've got a folder full of data, you can run a batch script on that data. And so I wanted to show you how you can think about building these general analysis scripts. And finally, I guess everyone's hearing about this deep learning, and we've covered this already in the workshop using open source tools, but we can also do this at the microscope. And I think this is a really powerful capability that we have, and something that, you should think about, taking advantage of while you're at the Zuckerman Institute. And this is the sort of three main components. And these are things that we can do using CSV Deep and other tools we talked about in the previous workshop through ImageJ. But if you're at the microscope and you want to do this in a sort of more efficient way, the three things we have is enhanced AI, which is really a method of imaging low signal to noise or very noisy images. Say like when you're doing live imaging and you really just can't afford to expose the sample with much light, or you need to image quickly, and then train the network so that when it sees this sort of noisy image, that it should really look like this really nicely exposed image. And so the training involves taking sort of paired data sets, one with very fast, noisy imaging and one with very slow, long exposure, high power images, so that we can train the network to interpret the noisy image and make something that's high contrast so that we can analyze that very easily. So that's one tool that we have. The second one is Segment.ai, and this is what we'll sort of go into today in terms of how you can train this, because within like a day, you could have this trained and it can really just transform how you're performing your analysis. And essentially what we're doing is teaching the network to say, okay, when I have this image, and it could be, DIC like in this case, or it could be fluorescence, I want you to have a binary mask where the axons and dendrites are, or I want you to really accurately segment these neurons amongst all the neuropil and branches and tissue background, which is very hard to deal with using conventional segmentation techniques. And finally, and this is something that's really powerful for when you're limited by the number of probes that you have, but you want to use a probe for some kind of tracking or analysis. So, the example here is where the network has been trained to, first imaging a DIC, transmitted light image, at the same time as having fluorescent markers. So, this could be like a marker for the plasma membrane and a marker for the nucleus. And once it's trained with that, you can just show it, okay, here's my DIC image, where would the nucleus be? And obviously you have to be very careful with these networks. You should really make sure you're using them exactly for what they're trained for. But people are using these for really, great things. Like a good example is cells where you're imaging live and you want to be able to really delineate those cells accurately, but the plasma membrane marker is a little bit patchy and very difficult to be sure about. What people have done is just train that so that when they see that plasma membrane marker, it'll know where the nucleus should be, and it will place the nucleus in that image so that then you can use that as a seed to be able to detect the cell. So that, if any of these things, if you're hitting a wall and you conventional analysis is just not working for you, come back to this and think about employing these tools. And this is something that we can definitely help you with in the platform. And hopefully after today, you'll be a little bit more comfortable even approaching that yourself. And so with that, I wanted to stop, and then we'll remote into the workstation at the Zuckerman Institute.
Can you just clarify quickly, look, the Elements, you were saying that we're using open source software mostly, but Elements isn't.
Yeah,
Elements
that's right.
So paid for, right?
Exactly. Yeah. So normally, that's why everything else has been open source, because I really feel like that's important in terms of sharing data and helping people who don't have access to commercial tools, or you yourself, if you leave here and you don't have Nikon, this isn't so useful for you. But in this case, because we're controlling a microscope, we're a little bit more restricted. And I also think that this is actually quite a powerful tool, and because we have so many microscopes using this at the Institute, that's sort of why we're doing this workshop today.
Okay, cool. And the jobs isn't also an add-on on top of the basic NIS elements. Is that correct?
That's right. Yeah. So we're sort of on top of elements. We have these modules. So jobs is one, general analysis is another, and the deep learning AI tool is another one as well. And so we have these. Those are on both the spinning disk and the slide scanner, where there's like the most use for them, and everything except the deep learning is on this new upright microscope that we sent an email out about recently. So
cool.
Yeah.
Thanks.
Yeah. Okay. So And bear with me if it's hard to read or you're having trouble seeing what I'm doing. Please just let me know in the chat or just shout out, because I know there's a lot going on in Elements and the font can be pretty small, but I'll try and make it easier to read. So this is the Elements installation on our spinning disk, and the tool that you're probably most familiar with using is this ND acquisition window. So hopefully you can see this in the center of the screen here. So this is where we can do time lapse and multi-point, large image, all of these things. But as you probably have felt, sometimes it can be a little bit restrictive if you want to do something more complicated. And so the tool that we can use instead is jobs. And in our case, we have these pre-built jobs just here in the jobs toolbar. And so we have ones for doing multiple slides and for imaging a slide, detecting sections, and then imaging at higher magnification. And just to show you what they look like, if I right click here, I can edit this job. And actually, like, maybe it looks a little bit complicated straight away, but it really is quite straightforward. I can just walk you through what we're doing here. So just to give you your bearings, like the jobs interface, over on the left-hand side are all the things that you can think about automating inside a job. And so we have here, things to do with well plates, which you can image automatically on the system as well. Different types of sample holders. In this case, we have that 4-slide holder that we use to automate 4-slide imaging very quickly. Things for time-lapse imaging, to deal with multi-points, which we can use to sort of build up, 3D montages and things like that. Large images to deal with, imaging tissue, all the stuff with Z-stacks, ROIs, autofocusing, perfect focus, which we also have, which helps to lock onto the cover slip. So if you're doing live imaging and you don't want to have issues with drifting, this is something we can also control through jobs. A lot of things with acquisition. And then because it's a scripting tool, we can do things like if or, loops and all this kind of stuff. So like I sort of mentioned previously, any kind of complex experiment where there's sort of several passes, like detecting tissue, then detecting a cell, and then changing objective, all of those things can be sort of thought about in here. And you can even get it to email you once it's finished if you want to go that far. So there's a lot of functionality in here. And in the case for our pre-scan job, all we're really doing is saying, okay, I want to capture an image and I need to tell it how I want to capture the image. And so for this first step here, we're saying use the DAPI wide field setting. So this is a special setting that just uses the wide field LEDs. It's a little bit faster and more forgiving for imaging tissue compared to the confocal. And then in the second step here, we're just saying, okay, where do we want to save this? Because we want to have a copy of this file. And then finally, we make a region list. This is just for the different tissue sections we might detect. And then we've created a parameter list just here. I can open this up. It's a little bit hard to see, but really we're just giving it a name where it's a choice. And the sort of options here separated by a backslash are just different options. So 1 slide, 2 slide, up to 4 slides, and then a smaller region. And when we run the job, we can select one of those options, and then it will use these sort of if loops to decide what to do. And so if we say, okay, image 1 slide, it's programmed in here to know exactly where that 1 slide would be, and then it just images that slide. And so you can see here, this is where we've marked out the corners of that slide. And then here is what settings to use to scan that slide. And so we're just saying, okay, use the 4x objective and just do it quickly. We don't need a perfectly stitched image. We just want this image very quickly. And doing this, we can image a slide in about 2 minutes per slide. So very quickly, in 8 minutes or so, you'll have a map of all 4 slides on the microscope. And we'll go through this step by step to build a job. I just wanted to give you just a quick walkthrough of one of these existing jobs first. From a job like this, we could get an image, and I have an example image that I can show you before we get started. So this is just an example of how we'd image a pre-scan section, and of course you could manually draw on this image, but if we're using something like our AI detection, this is up here. And we can use, so these are those options that I was talking about before. So we have enhance AI, convert AI, and segment AI. And if we wanted to detect a tissue here, we can just say segment AI. And we can tell it which tool we want to use. And on here we have a couple of different tools already sort of pre-trained. And so we could use this one for tissue segmentation. And you can see that it's already sort of detected these ones here. And of course, when we use this in a pipeline, we use a little bit more sort of control, so we can filter based on size to get rid of some of these smaller features. And we can also sort of filter on circularity so that we can get rid of any kind of very linear things like the edge of a cover slip. And so these tools are much better than using just standard threshold and things like that, because they're more robust. So we've trained these on very low intensity, very high intensity, no labeling of just neurons rather than a global marker like DAPI. And so that sort of really helps you to use these sort of segmentation and imaging tools in a more reliable manner. Because one of the most frustrating things that comes up when you try to do this is you get something working really well for detecting your tissue and detecting the cells. You come with a slightly different experiment and it's just all completely broken, nothing's working, you have to rebuild it. And using these like segment AI tools gives you something that's like far more robust and reliable for doing this type of segmentation. Let's actually start building a job from scratch. So we can do this down on the bottom right corner, we have this Jobs Explorer. And I can create a new job just by clicking here. And just shout out if you can't see the text clearly or anything like that. So the first thing that we want to do is tell it, okay, I want to pre-scan this tissue section like we sort of were talking about before. And so in here, we can go down to our acquisition. So right down here in acquisition, we can set up a capture definition. And we can tell it what this is going to be. So this will be our pre-scan capture definition. So we can call these something so it's easy for us to find later. And we also want a definition of how we're going to capture this at higher magnification. So we can add a second one here. This is going to be our scan definition. And I'm just double clicking them to expand them or shrink them. And I'm going to pre-select something. Maybe in this case, we'll have DAPI, and on this slide that I have on the microscope right now, we have some good labeling with Alexa 647. So we're going to image with 2 channels here when we scan finally. We also want to make sure that when we're scanning that we're auto-focusing. We don't want to image these sections and have them out of focus. And so what I'm going to show you today is just a simple autofocus. This is where we'll find the middle of the region and then we'll focus on that. But there's, and this is fine for small tissue sections or when things are well mounted. But if you have something where it's very uneven or very large tissue sections, then we can do things that are more sophisticated, like making a focus map where we actually find points across the tissue and interpolate between them so that we can create a really nice map of where to image across the section. And so that's something we can help you with, after this workshop. And if you're ever trying to sort of understand these autofocus settings, just make sure that, if it's fluorescence, use a fluorescence criteria and make sure you're using an OC, an OC is the configurations that we're imaging with. Make sure you're using something that has good contrast. So I know DAPI is something that's very easy to see on this tissue, so that's something that I can rely on. And we're using the autofocus mode here to just explain this. A single pass, it will just go through that range with a set number of images. And 2 passes is a nice way because it will sweep through with a very coarse focus and then figure out where the best plane was, come back and then sweep through just around that plane at a much finer step. And that way you'll have a good chance of very quickly finding a plane that it's worth focusing on. Okay, and so we also need to have a region list. This is if you're ever doing anything where you're imaging and you're detecting an array of different regions, the jobs will need somewhere to store those different regions. And the way you can do that is underneath large images, there's an option here for region list. This is especially important if you're doing like automatic detection of the tissue sections. And we also want to make sure that we're using the right objective. So, maybe you know that you're already going to be on the 4x objective, but perhaps you just want to make sure that the microscope will already be on, no matter what objective you start on, it's going to go to the 4x objective. And to do that, there's actually not a tool that's inside the jobs modules available. But that's okay because there's a whole lot of functionality in Elements that's not in jobs, but we can get to through its internal like macro scripting functionality. And I can show you how to do that. So in here, there's a macro tool. And you may be like, oh, I'm never going to remember what macro is to type in or what to do here. Nikon have actually made this very easy. So this function explorer button here, if you click on that, it'll let you search for all the things that are macro recordable in Elements. And they've done a fantastic job with this. Almost everything that you do within the software is callable through a macro. So if you can't do it through jobs specifically, then we can do it through macro a lot of the time. And in our case, the objective turret, I'm just typing in nose piece here. This is the objective turret. And this is kind of similar to what we looked at in ImageJ macro. So you can always see like set, that's saying do something, and get is just saying asking like something. So get would say, okay, well, what is the objective that's currently in place? And then set would say, okay, set the objective to this. And in this, we're just saying set the objective to a particular position. And the position on the nose piece, so this starts at 0. So 0 is 4x, 1 is 2x, is 10x, 2 is 20x. So that's how you can think about it in terms of the objectives we have here. So this will set it to the 4x objective. And then we want to scan this pre-scans of this whole slide or multiple slides. And so to do that, we want to go to large image. I'm just going to place this below here. Actually, in some of these cases, we can call the objective specifically. So in this particular module, you might not even have to run this macro. And Elements is sort of always improving on this. So you may find an easier way if you come to this in a couple of months when we update the software again. But for now, like we can do it here as well, and we can set the number of fields. I just want to use a very quick example today. So I'm just doing a small like 6x6 area. And whenever I'm doing something that's quick, like a pre-scan, just to get my bearings, I don't worry about doing precise stitching, and I don't worry about overlap. This is still going to give you a nice image that you can use to delineate sections and things like that. And we'll come back to this actually. If we want to keep this image, we have to have this on. But if we're doing something like we're imaging the whole slide and then we want to image the tissue sections afterwards, sometimes you can't do both. And so in our case, we actually want to keep the images of the sections rather than keeping this pre-scan image of the whole slide. And so we need to switch this to never or later, although there is a way to get around this with general analysis. So we may look at that in this workshop as well. Okay, so we've got this set up nicely. For the first example, I want to keep it simple. And so rather than doing some automatic detection of the tissue, we can just do something where we draw the regions. And this is still a little bit nicer than using ND acquisition, because with these draw regions, you can do very quick rectangles, or you can be more precise and use a polygon tool to delineate these tissue sections more accurately. And there's nothing we really need to worry about in here. It's just we just need to make sure that it knows what image it's drawing these regions on. And so this image is called ally scan, and so it's saying, okay, we're going to draw these regions on the ally scan image. Okay, so that's what we have to do here. Once we've drawn those regions, then we want to image them with a different magnification. And so in our case, we can just copy and paste our macro here again. So this, we can even call it something so it's easy for us to see. And we know that this is position 0, 1, 2. So this is the 20x if we put 2. And then we want to do a loop. So we've scanned with the 4x objective, we've drawn these regions, and now we want to go and image each of these sections. And so in the large image list here, we have an option loop over regions. So we can bring this in, and now we can tell it the sort of things we want to do for every region. And so one of the things we want to do first is just move to the center of a region. So we can put this module in. This will just take us to the center. And so if you're imaging like brain tissue or something like that, that's probably going to be a good spot to focus. And then we want to do our autofocus. So if we go back up towards the top here, yeah, these can be, I don't think there's a search option for these. So you just have to be patient sometimes and look through this list. But first we did the autofocus settings up here. Now we're just running this autofocus. So this will just do whatever the settings are here. And if you wanted to have a setting for autofocusing on a slide and then a setting for autofocusing on the tissue sections, you can have multiple sort of sets of these autofocuses. So you're not limited in that way. And then finally, we want to image those regions. And so in here, we're using a different one now. We're actually scanning the ROI. And so this ROI might be sort of amorphous. It might not be a rectangle, and we don't want to waste any time imaging a full rectangle if we really only want exactly where the tissue is. And that's what this tool allows us to do. And it's saying, okay, we're going to use okay, so it's saying it's going to use the pre-scan setting, but we don't want that. We want it to actually use the scan definition. This is our different settings that we made up here. And this is the objective we want to use. And we want to save these images. So that's all we have to worry about here. Yeah, and just to make sure as well, like here we had this region list, which is useful when you're doing automated imaging or automated segmentation. But for draw regions, you actually don't need to have this. So we're just making sure here that it's going to look at the draw regions list, this one here, for imaging each of these tissue sections. And then finally, we just need to tell it where to save this stuff. So there's an option, I think down towards the bottom here for storage, just here. And we just want this at the very end, outside the loop. And if we run this, if I click play right now, it's just going to get straight into the imaging. But we want to make these a little bit more user-friendly. So if you're doing something like this and there's some things you want to change different times when you're at the microscope, then what you can do is any of these that are important for you to change, you can right-click and say put in wizard. And so anything that you're planning to change, maybe you want to change the way that you're scanning sometimes, or maybe you want to do a pre-scan with GFP instead of DAPI, then these are important ones to put in there. Maybe you also want to put the autofocus in, because maybe sometimes you won't have DAPI and you have like deep red neurotrace, and that would be a better thing to focus on. So anything like that, put them in the list. You can put the storage in the wizard list as well. And that way, I'm just going to save this. When we run this, we'll be presented with a workflow that we can start to sort of answer these questions and get something working. And so whenever if you've used the system already and you've used some of the jobs we've made, this will be very familiar to you. But if you haven't, you always just work from top to bottom on this left-hand side. So each of these tabs you just need to check through, and they'll be marked green after you've looked at them. So we know this is fine. We know that this is fine. This is the settings we want to capture with on the 20x. These are our autofocus settings, which I think will be okay. And this is our storage location. So I can just give it a name and I can tell it where to save the data. Actually, just before we do that, I just want to check to make sure we have something that we're able to look at. Okay, so we have a live image here. We're in the middle of the tissue section. So if we run this, and if you can't see what's happening, we can just say auto scale here. This button here will just scale to the brightest pixel in the image. And you can see, with the 4x objective, even without any overlap, like we're getting a nice image here where we can really clearly see the tissue. And because we're imaging with wide field, even if it goes out of focus slightly, we're still going to be able to see the tissue quite clearly. And if I move this to the side, this can be a little bit awkward sometimes because this is the image that was captured. I can't see what's happening in it. I'm just going to move this to the side. Maybe I'll move this. I'm going to click auto scale. Okay, so if that's not working, let's just see. This is our lookup table down here, so I can manually, just in the bottom middle of the screen, I'm just going to manually adjust this so I can see the image. Okay, so hopefully you can see the image okay. And one thing to point out, this is where we can draw our regions. And this is where we click finish when we finish drawing the region. So there's also a finish button here, but you don't want to press this, otherwise it will just stop the whole job. So if I move this out of the way, I can draw a region. And I'm just going to draw, rather than draw this whole tissue section, which will take a little while, I'm just going to draw a small region here. And if I wanted to maybe image another region, then I can just draw another one here. And when I'm happy with that, I can click finish. And what the job will do now is image those separate tissue sections. And so this is auto-focusing, so you can see that quick sweep through, and then it's coming back, and now it's doing a finer focus just where the tissue was found, and now it's imaging. And this is just there's some sparse labeling of some neurons in this brain section in far red. Okay, so it's as easy as that. So we've got now a clear image of these cells just by using the jobs tool. And of course, you could combine that with the pre-scan, the sort of 4-slide pre-scan that I showed you. We could build something like that and follow it with something like this, and that will save you some of the playing around in the ND acquisition tool. And I guess the sort of maybe some of you are thinking about, okay, but I want to do that in 3D. That's also possible. It's a little bit too lengthy to go through in this workshop today, but we have those jobs built on the system, and I'll also upload these to GitHub after the workshop. So along with the recording, there'll be some of these working jobs for different applications, so that you've got a good starting point for some of these more complex jobs. So the two things that you might want to add to this are, okay, how about I automatically detect the tissue rather than using these manual regions? And that's something that's definitely possible. And the other thing is, maybe I want to count the cells at the same time. So after I get these images, what I was planning to do is take them back to the computer, and sometime later on when I have some time, I'd have to put them through ImageJ and have a pipeline where I could count those cells. And that's a whole nother sort of thing that I need to think about. But if I really know that that's what I'm planning to do with these images, one option you have is to really just set it all up within the job. And this is coming to the general analysis that we talked about in the talk just a few minutes ago. So I'm just going to minimize these. If I come back to our job, so if you were going to do automation in either of these things, detecting the tissue or detecting the cells, then we would put a general analysis step in. This is towards the bottom here. And we're actually using General Analysis 3. So this is a slightly newer version of their analysis processing tools. And instead of drawing regions, we would put it here. And then we would have to make a job. We'd have to make a new one where, okay, it would have the, actually, I can just very quickly show you. I don't want to spend too much time on this, but if we had our, say this is the image that we're getting from our pre-scan, then we can make a new general analysis here where we say, okay, this is the image that we're getting from the microscope, and we want to segment it and then pass those regions on so that we can scan those tissue sections. And something simple like that is actually quite easy to build in here. And the good thing about general analysis over jobs is they have a search. So if you're really having trouble finding things, you can just search for them and find them. Actually, before I even get into this, I can just walk you through some of the things that are available to you, because this is actually a really powerful tool. Each of these tabs here have all the different modules for performing image analysis. So pre-processing is anything you do to the image before you start making measurements. So these are all filters that you can use to make it easier to isolate the cells or detect edges or anything that might be difficult if you were just planning to segment on the raw data. These are processing steps like creating a maximum intensity projection, things like that. Image operations is where you might have a background image. So maybe we have a flat field image for the microscope that you want to subtract from the data you're acquiring to get really nice clean data. That's something that you could do in the image operations. There's some of these conversions, like some of these will be familiar to you from the macro course we did, like on merging and things like that. And then all these red ones here are to do with the segmentation. So segmentation is, and we'll go through this in a little bit more detail in a minute, but creating a threshold so that you can find the cells or the tissue sections. We'll go through using bright spots, which is a really nice way of isolating individual cells in a conventional way. And once you have those sort of binary masks, you might need to do something with them. Maybe they're really tightly packed and multiple cells are looking like just one cell. And so that's where you need to do some binary processing. So this is where we can erode the masks so that they're more separated, or split touching objects, things like that. So that's in binary processing. And then this is kind of a little bit more advanced. Maybe like you have multiple channels that you've detected, so a red channel and a green channel, and you want to ask like, okay, only show me the masks that are overlapping, or maybe there's some co-localization or something in a cell that you need to get an idea of. So these are sort of operations using multiple binary masks. And then of course the most important thing is getting some measurements. And so we have the ability to get all the sort of measurements that you're interested in, and then build those into a spreadsheet that you can export as a CSV file. And you can really customize exactly what you want to extract, like the number or the intensity, the area, all those sort of things. And we will go into doing that at the end of this session. And of course, because this is a spinning disk confocal and many of you are working in 3D, these tools have actually also been extended to 3D processing. So this is something that is a little bit slower and heavier to sort of do. So if you're planning to do something like that, I would more think about capturing the data with a job and then running a batch processing afterwards to sort of minimize the sort of time that you're at the microscope, reduce the cost of the experiment. And these yellow ones here are all to do with getting the data out. So making that CSV file and naming them in a sensible way. And I'm going to share some methods for doing that at the end of this workshop. So just getting back to the example at hand, we have this fluorescent channel and we want to detect the tissue sections. And so the way that we can do that, we can use our segment AI that we've already trained. And I'm going to show you how to train this too in the workshop. Okay, so it's in here, it's this one just here. And the way that this works is it's quite intuitive actually. These just connect together like nodes. So this segment AI module is asking for an image or an input. And to do that, I can just click this node and drag it onto the channel that I want to process. And straight away, it will try and process this. You can see there's an image warning here saying, look, this image is too big. What we can do in this situation, we can force it to do it anyway, or we can just create a preview ROI. So I can just say, okay, look, I want to quickly troubleshoot this pipeline. I don't want to be waiting to process the whole thing. So I'm just going to make a ROI. Maybe I'll do it like this. And then right click to lock that in. And now you can see it's thinking about what the segment AI will do to this image, and it's showing us the masks. That's kind of all we have to do. I mean, one nice thing that we do sometimes to make it easier to see these different regions, we can color by ID. And that way, like the tissue sections will have a different color. That's nice if you're doing really dense cell labeling and you're not sure if cells are splitting or still touching. Okay, so it's as easy as that. If just for example, this didn't work as nicely as it just did, in here we have a few limited options we can use to make sure this output is a little bit cleaner. So maybe the AI is doing a good job, but not a great job. Kind of like when I first showed you this at the beginning. Then you can use these additional tools like smoothing the data a little bit, maybe put a size filter in. Like we know that these tissue sections are all going to be bigger than, a few thousand microns. So there are things that we can add to make this more reliable. And if we were still having trouble, we could add filters in beforehand or add some binary operations afterwards. So don't feel like if this isn't working, that's the end of the story. There's always a bit of room to improve it further. And that's basically it. So we can save this as a new general analysis. I can close this. And now it knows, okay, scan the slide, use our general analysis we just wrote. And it's really important if you do this, that you update this region list tool. This is something that causes a little bit of trouble right now in Elements. This tends to default back to these settings if things are changed inside the job. So if you're ever having trouble with this, these are the settings you need to update. You need to tell it that when it detects these tissue sections, put them into the list. So that's our list up here. So just put them in the region list. And we don't want to append them, we want to replace them. Appending, if you're doing multiple slides and things like that, it can start to have some trouble. So replace is what works best here. And per object is, so if you remember these three, then the output you'll get from this is a series of regions that can then be imaged in our loop down the bottom here. So that's how we could get this to work for tissue sections. If we wanted to analyze the cells, then what we can do is do another general analysis. And I'm putting it in at the end of the loop. So after it moves to the center, it will focus, scan the area of interest, and then do the cell count. And we need to make a new job for this. So I'm going to create a new job here. And whenever you're doing this, make sure you have the image that you're analyzing open. So it's a little bit confused right now because I have this DAPI image here. So I'm just going to close this. And this is the image that I'd like to work on because it's reflecting the analysis that we plan to do. And maybe like so I'm just cropping the image. I don't know if you saw that there's a crop tool just on the far left here. And you can just draw the region and then click OK, and this will create a new image. This will just be a little bit faster for us for troubleshooting. Okay, so now if I create a new analysis, it knows that we have these two channels to work with. And the first thing we might want to do, actually, let's just look at this channel by itself. One thing that, we could try and just use a threshold. That can be a little bit tricky sometimes because there's cells here that are very bright, there's cells that are very dim. Actually, let me even make this monochrome so it's easy for you to see. Oh, if it'll let me. Yeah, let me just change it so it's easy to see. If you're ever looking at, an image in color, it's a good idea, especially if you're trying to make sort of measurements or interpret it accurately, to at least switch it to monochrome, because you don't have a very good ability to perceive intensities and contrast in certain colors, especially blue and magenta. So having it in grayscale will allow you, at least, as best as you can, see the different intensities in the image. And I think we're cutting some intensity off here, so I'm just going to drag this all the way to the left so I can see the background if there isn't. Okay, it's just that those cells are very bright. Anyway, try and get an image that's easy to see before you start making adjustments in an analysis pipeline. So now we have this image. If we're going to try and detect these cells, if we just use a standard threshold, we could run into some trouble. And so one thing that we can use instead is just try and make the individual nuclei here homogeneous, and then use a tool that will find the centroids of those cells. And one way that we can do that in this software is to do some image processing. So in here, we've got a whole bunch of filters. And if you join some of the previous workshops, we talked a little bit about how to use some of these filters. So in this case, I'm going to use a median filter. And we just want to choose a radius that is going to make the nucleus or the cell homogeneous without really doing anything else to the image. And so in this case, something you can actually, there's a good way. The good thing about this software is you can very quickly switch between views. So see this little node here, if I want to look at what the unprocessed image looks like, I can just click here. That's showing me what the raw data looks like. I'm just holding down Control and zooming in a little bit so you can see more clearly. And if I click on this one now, it'll show us the filtered one. And so this median filter, it just knocks down any peaks that might be inside the cells. If we had multiple peaks inside the cells, then we could get multiple counts inside a cell. So doing a median filter just like flattens that out. And what we also might be worried about is maybe some background in the tissue. And so something we talked about a lot in the previous workshops is a background subtraction. And we can do that just here. So I'm just connecting this onto our median filtered image. And right now, this default setting is clearly too small. If you go too small with a rolling ball filter, you'll delete things. So we want to go a little bit bigger so that we're preserving the cells, but we're getting rid of any kind of lower frequency information that's in the image. And there's a good explanation for this in the ImageJ workshop if you're interested. So we can quickly, this is actually one of the great things about this workflow is you can very quickly go back and forth between images and see what you're doing to the image and see where you need to modify things. And so it's actually a really nice way to build out analysis pipelines. I'm not going to spend too much time on this, but I feel like we're getting pretty close now to the right setting here. And now what we can do is use a segmentation tool. And so if we go to segmentation, bright spots is a good one for this. I'm just connecting it to the filtered image. And if I click in here, I'll be able to see the options for doing this. And right now we haven't got a threshold for the intensity, so we need to come up a little bit. I find the circular area is a little bit hard to look at, so if I switch this to spot, it's going to be a little bit easier to see. And then hopefully it'll let me bring this intensity up. I can make the spot a little bit bigger so that it's easier for you guys to see as well. So you can see this very quickly. I mean, if you don't do the background subtraction, that intensity could be very hard to figure out. But because we've done the background subtraction, outside of these cells should be very close to zero. And I don't know if it's going to let me show you. Yeah, just down the bottom here, if you can see, when I hover the cursor, it's telling me what the intensity is. And so the intensity, there's two numbers in brackets. The first one is the DAPI channel. The second one is the far red, or the Alexa 647. So by hovering around, they can see all these pixels are basically zero. And that's because we filtered the data and then we did background subtraction. And so that the background is actually quite homogeneous, and then the rolling ball filter has just removed that out of the image. And so we have like a really nice high contrast, like straight from zero into the, oh, sorry, I have light. So the far red there is around 3000 now, and out here it's like two or three. So very close to zero. Okay, so if we're happy with that, the nice thing about this is we're finding the center of these cells, but if you still want the area of the cell, you can get it. You can just tell it here to grow the region after it's found the cell. And we have to be careful here that we again have some kind of threshold of intensity. But the nice thing about this is because you've found the cell and then expanded outwards, you have less of an issue with touching objects. So if you just did a threshold on an image, and a good example would be these cells here or these cells here, they're going to look like one cell, and you're going to have to rely on a watershed to split those cells. And if you ever use a watershed feature to split cells, in some programs it's very unreliable. Like if we ran it on this, then probably we'd end up with two cells here, even though there's one cell. And so using what's called this sort of like seeded object detection is much better, because we can find all the cells and then we can expand their boundaries to the edge, and we can be very clear about how many cells there are. So, I mean, if this was like a real project, I would spend more time refining it, but I think just for the sake of getting through this more quickly, I'm going to leave it at this. If the cells were a different size, of course, we could use a different diameter here. So this is really important for finding the cells, the intensity we talked about, and then the threshold for the region growing we also discussed. So that's all we have to do. So that's working for detecting these cells. I know, of course, we might want to color them so that we can see them more clearly. Oops. Yeah, and I encourage you to explore this too, because there's so many features in here that, if you're stuck on what to measure, you can always reach out to us and we can like give you a good hint on what tools are good for measuring different things in images. But yeah, it's worth exploring what you can do here. Okay, so we've got this looking nice. Maybe we also care about, actually getting some information out of this. So how many cells or what the intensity of the cells are. And so to do that, we can get a measurement. And, the basic ones here, like we can do an object count. So we have to, when you're doing these, you have to connect them to a binary image. So these two here in green, these are binary images. These are 16-bit images. So if I tried to connect it, and to unconnect it, I can just double click. If I try and connect it to a intensity image, it just won't connect. So I'm going to connect it to this binary image here. Then I can see straight away how many cells I have. I can, so also, it starts to get a little bit busy here as we build things. So you can hold down Shift and drag if you need to select a couple of things. That way you can move them. Maybe we want to move them here so it's more of just a vertical process. And maybe to make room, I can just select this. I'll bring it all the way back up here. So we start with the analysis side of things on this column. Okay, so we have our object count. Maybe we also want the total number, or actually the mean intensity. This is a good one. So I want to know the mean intensity of all these. Okay, so here, this is our intensity. So if you hover over them, it will tell you. This is our intensity. So this we should measure on our raw data. So I'm just going to connect it to our raw data. And our binary, I'm going to connect onto our binary mask. And if I yeah, we want mean object intensity. So this current one is just measuring the sum of all these different ones. We actually want like a per object. So actually all the per object ones are object shape. So yeah, be careful sometimes with this because there are so many modules. And you can just delete these by pressing delete. I'm going to do shift and select to just move this over. So again, I'm just, this is connecting to the binary. This is connecting to the channel. Okay, and so now I have something that looks more sensible. This is my list of objects, and it's telling me the object ID and then the mean intensity of those objects. If I wanted to get something like the area as well. Yeah, so definitely use the search for this because there's a lot of functionality. Okay, so now I have two records. I have three actually. I have the total count, I have the mean intensity, and I have the object area. If you were trying to do a project where maybe you wanted to standardize the mean intensity to the object area, or are you really interested in both of these measurements, rather than just making two spreadsheets, we can combine these together. Yeah, join records. So here we can just combine these together and hopefully, yeah, okay, so now we have our intensity and object area. So this is one table that we want to just get out of the pipeline when we run it. Okay, so this is actually pretty good. The next step is actually way more complicated than it really needs to be. We want to actually get a CSV file from this, and if we try to run this on multiple images, if you try and do this later on, you'll find an issue where it just tries to write the same file every time. And so what I've got is a pre-made job that does this for you. I'll actually take you through it so that you can see how it works. But I would just say I'm going to upload this to GitHub, and if you want to build something like this so that you're automatically imaging and analyzing cells and things like that in Elements, and you're having trouble with this, just reach out to us because we can help you with this very quickly. I'm just going to save this just so we have it saved. So I actually set one up previously, saving the CSV file. I'm going to walk you through it anyway, just so that you can have an idea of what's going on, but it's not something you really have to remember how to do because it's just a little bit more complicated than it needs to be. Let me just show you. Yeah, so any of these analysis scripts, you can just select them in the drop down. These are the ones that we've already built before, and you can edit them here. Obviously, like, don't edit the ones that the platform has set up, like make your own so that you don't create any issues for people who are using the instrument. This might look a little bit different, but it's actually the same job that we were building together just then. Like, you can see the filtering, the bright spots, the object filter, and the measurement for intensity and area. I'm joining the records. So all of this stuff, I'm just going to move it to the left. This is all the things that we already talked about. So don't get confused by this. And the final step we're doing is exporting it to a CSV. So this is just a simple, let's put this in the center of the screen. This is just a simple tool here in results called, underneath reference, export table to CSV. So CSV is just a very simple spreadsheet that you can open in Excel or many other programs. To save it as a file that we can interpret, we have to add in all of these modules here. And all this is doing is getting the input folder, or the folder that we're capturing the data into. So this would almost be called like the output folder, really. The file name, so this is coming from the image that's being captured by the microscope, and some input text. And we're just putting a .csv at the end so that it knows to call it a CSV file. And these steps below here are basically modifying the column so that I can show you. So we're just getting the file name, not any other information. And then it runs like a little bit of JavaScript in here to basically get the input folder, get the file name, get the CSV and put them all together into a single string. And in fact, I've just modified this one here to put a timestamp on the file because sometimes because jobs are still kind of under development, we don't always get a file name by doing this. So in any case, if you're using this, let us know and we'll add this in for you. And what this means is when we run the job, you'll get a file in the folder saying how many cells were in each image. And so that way you can get an immediate analysis done. And I can run this on the fly for you now. It's always a bit risky with live demos, but I think we can get this to work. So what I can do is say, okay, use this. I'm just going to get rid of this one. And it's complaining here because it thinks, so right now it's saying, look, it's not compatible with the data that we're generating. So if you see something like this, just have a think about what it's looking at. Right now it's looking at the pre-scan image, and it knows that that is going to be a DAPI only image, and it's been trained to work with this DAPI and far red image. So I just need to tell it, okay, look, use this region image just here for doing the analysis. So that's how we can think about that. And outputs, actually think about what you actually want. Like, you're already getting the image anyway when it's doing this capture here, so we don't need that image again. Maybe you want to look at what maybe you want to validate the pipeline and see how the binary mask has appeared over the cells. And so in that case, maybe you do want to leave these on for a little while to make sure it's working, or just keep the binaries, which will be a smaller file. And certainly we want to keep the tables because they're going to have the cell number. We don't have to worry so much about these other ones here. So this should work. Actually, I'm just going to put this in the wizard as well, so that I can set a directory for where to save those CSV files. And then I'm going to save this and run this. And just like before, I'm keeping this the same. I'm just imaging DAPI with wide field, DAPI and far red for the 20x acquisition, focusing with DAPI. I want this, actually I'll just save it to the desktop, that's fine. And I'm going to, actually I'll put this to the desktop too, that way we can just quickly see. So normally you would just save this to the data drive and to your own folder. And so right now it's switching back to that 4x objective. So even though I'm actually running this outside of the institute, but we're still getting something to work. Here's our pre-scanned image that we had before. And then just quickly again, I'm going to adjust the lookup table here just so I can see the tissue. And then I'm going to draw some regions that I want to count in. And then actually, before I do, we were talking before about using this deep learning tool for detecting the tissue, but there's also like the way that that's trained, it can be quite good at detecting features that are have different texture or are visually different to surrounding areas. So what we haven't tried this yet, but I think one thing that you could do is, if you're really interested in imaging just the hippocampus, of course you could train it to do something like this and then draw around the hippocampus with these ROIs. But it'd be worth trying to train the network to detect just those areas, because if they're visually different enough that you can see that difference, then the neural networks are quite good at, if they're trained specifically for something like that they can perform it. So if you are doing a project like that where there's something visually clear that you want to isolate, it's worth investing a little bit of time, and I'll show you how to do that in a minute, to train a network and potentially have something that's very automated for doing something that would otherwise be very time consuming. So I'm clicking finish so that it goes to image those regions. I'll move this back so we can see it. And we're doing the autofocus now. And then we're doing the tiled image. Okay, so these are our images, and it's running the analysis as well. So at the end there, it's just doing the cell counting for these images. And if we look on the desktop, when this is finished, we should have those CSV files that contain the cell counts. Okay, and then these are those files here. So these will appear like I would normally save them in the same folder that has the image files. That way there'll be a file for the image and there'll be a file for the CSV file. We can, so here's our object intensity, our object area that's ready to plot in whatever tool you use for plotting and statistics. And here is a image that shows us how well the segmentation worked. And I would always encourage you to get these images before you trust a pipeline. You can see that actually there are some cells that we missed during this like there's some cells here. And you can toggle these on and off just over here. So it's done a pretty good job, but there's definitely like some areas there that I might want to improve upon. And that way, having these images as feedback, I can start to work on the pipeline a little bit more. But you can see it's done a pretty good job. I mean, as long as you sort of compare this to a manual count and get an idea of what the false positive, false negative rate is, then you can have a good idea of how reliable your cell counting is. Okay, so the very last thing I wanted to touch on is using Segment.ai. So, this is actually an example where the cell segmentation is relatively easy. And I think with a little bit more work, we'd be able to get it working very reliably. But, if you're working in biological samples where things are a little bit more complicated, and a good example is, in neuroscience, when we're looking at labeled neurons and we're seeing the axons and dendrites as well as the soma, it can be very challenging to get something to work well using just classical segmentation tools. And so if you want something that's more robust, then the Segment AI tool is a really good way of doing that. So they're actually very easy to train. And this is, if you want an open source way of doing this, then I would refer you back to the workshop we did previously where we went through using CSP Deep to do just this. But in Elements, it's very easy to do. I've got some example images here that are the kind of images that you might have trouble with. So this is an image in a brain tissue section. We have cell bodies, we have a lot of axons and dendrites, and we can try and filter this to really isolate the cell bodies, and we could probably get halfway there. But if we want something we can really trust, where we know we're getting at least 90% of these cells and not getting lots of false positives, then Segment AI is a really good way of doing that. And the way you would go about capturing data for a pipeline like this is, use the settings that you would be using for the acquisition of your experiment. So don't capture data with a different objective and then train it and then try and use it with the objective that you've decided on later on. Figure out exactly how you want to run your experiment, if it's going to be with the 10x or the 20x, and with binning or not. Figure all that out first. And then you can capture some data, and you don't actually need a lot of data for these networks. So this is a proprietary network. I don't know exactly what the architecture is, but functionally it's quite similar to the CSP Deep denoiseg pipeline. So you only need about 10 or so images to get a good result in terms of segmentation. So you don't need to capture a lot of data to start to get a reliable output from the network. And so you would just set your acquisition settings up. You could use a job to acquire that data, or you could very simply use ND acquisition to mark out some points in the tissue to acquire some images. And importantly, you need to have all those images into one data set before you start training the masks and then training the network. And the way that you could do that is if you have a variety of images. Let me just show you a good way to do this. You can go to File, Import, and then create an ND file from a file sequence. So these don't have to be, images from the Nikon microscope either. If you just want to use the NIS-AI for doing this, and you find it easier than using ImageJ or whatever it is, and you just want to do it this way, you can bring TIFFs in to do this as well. And so in my case, I have the files in here. And I would just define it manually and use a multi-point is fine, so that each of these is just sort of images that are connected in the file, but they're not like connected over a time lapse or anything like that. So multi-point is a good way of doing that. And I'm just going to say that there's 3 images because I want to save a little bit of time today, but obviously 10 to 15 would be a nice number. And that's it. I can click convert, and I'll end up with an ND file that has those images as separate points in this series. And I would try and capture data that encompasses the full range of image data you would expect, like areas where you have lots of axons and dendrites, areas where there's like some bubbles and things like that, so that you can really train it to ignore those things while focusing in on cells. And then to start to train to create the masks for training, let me just zoom in so you can see clearly. Just adjusting this so I can see a little bit better. With elements, if you right click on the background, there's some analysis controls here. And the one that we're looking for is this binary toolbar. And this is a really useful tool. You can try and detect all the cells at once if you've got a pretty easy job. Probably you won't be able to, otherwise you wouldn't be using the Segment AI. But just so you can see what this looks like, if I click on a cell with this multi-region selection, it will try and find all the cells of a similar intensity. This is not going to be so good because you can see we're already introducing some false positives. I would actually spend the time to carefully annotate this, because what you get out of the network is as good as what you train it with. And so if you start to include pixels that aren't really cell bodies in the training, you're going to have those kind of things appearing in the output. And so it really doesn't take any more than half an hour to an hour to sit down and really carefully annotate 10 to 12 images. And I would use this single tool, or the single sort of region tool, left click, and then I'm just scrolling with the mouse wheel to sort of adjust that threshold locally, and then right click to mark that. And this is just one way of doing it. You could be more strict and just marking like a center point for the cells if you're worried about touching objects. But you can see just doing this is quite a quick process. And I would just go through and find all the cells. And this is going to be, again, like there will be some personal like subjectivity on what you consider a cell. But if you're this is something, if you were manually doing, could fluctuate day to day. Like today I might say that that's a cell, but tomorrow when I'm counting I might be feeling a bit different, or the screen might look a little bit different and I might not count it. And so at least if you sit down in one session and train this in a very consistent way, the results that you're going to be getting are going to be consistent and reflect that. So I think it's still a much better way. So this is what I would do. An important thing to remember is it needs to have mask information on every image. So if I had an image where I just had projections, like this is potentially a good example. Okay, here we have like at least one cell. So if there was no cells here, what you could do, and this is what I've done in the past, is you could paste into the image some cells so that you're sort of artificially adding some cells so that it knows, okay, like this is some cells and this is what I want versus all this other stuff which I should ignore. That's one way that you can get around this problem. But just remember, if you have an image in this series that doesn't have a mask on it, then it won't run the network and it won't train the network. Okay, so I just need to do one more in this. So obviously I would do all the cells, but I don't want to take up your time doing that. So I've just at least marked every single image with some masks. And now I can go up here to this AI tool. And we're training a segment AI. So here we can train segment AI. We can tell it that it already knows a lot of this, so it knows the channel we're working on. We have our binary mask already. Actually, one thing, just before I move on, if you have any trouble with this, or you need to make some really fine adjustments, you can use additional binary tools by just pressing tab. So if you press tab, it turns on these binary editing tools. And a really useful one is just to use the pen. And here I can change it from foreground to background. So if this mask was pretty good, but I really just wanted to make sure that these 2 pixels here weren't part of the mask, I could just turn it to background and just paint to get rid of them. So do as much as you can with the fast tool. If you need to refine it further, then come in and use the paintbrush here by pressing tab to get to these options. And then, yeah, just spend that time to make it as good as you can, because you're going to spend, a couple of hours training this network, and you may spend, several years using it. So it's worth making that little bit of extra effort. Okay, so
But for every fluorophore and every combination of different labeling you use, it's going to have different background. You'd have to redo this, right?
not
It's not just
necessarily.
if you have cells of this kind, it would always work, or
Yeah, so I think like you obviously would want to check this, but if you had another neuron population with a similar expression pattern, like here where we have it in the cell body and the axons and dendrites, it doesn't matter what color it's expressing, this network will work well. So, the network that we have trained for working on tissue sections is robust across many different labels, and at least for the cell bodies in neurons, if they're virally labeled neurons, it will work quite well for that. But yeah, obviously your mileage will vary, so I would always check to see what output I'm getting. But yeah, it's more looking at, it's obviously looking at intensity information, but it's also using a lot of spatial information in the image, so texture and things like that. And so yeah, it's fairly robust to things like that.
okay, cool. Thanks.
No worries. Yeah, so that's it here, and then we can go into train segment AI. And actually, one nice thing I should mention is if you've trained a network, so training these networks, you probably spend about an hour to an hour and a half doing the masks. Training the network takes about 8 hours overnight on one of these systems. And obviously, let us know if you're trying to do that, and we can work out something in terms of, we don't want to charge you for the full rate of the instrument if you're just training a network. So let us know if you're doing something like this. And if you have a network that's working well, so this is actually coming back to your question, if it's working well for a bunch of neurons, but you've got a new label that's slightly different and it's not working quite as well, but it's pretty close, you can just label a new set of images with this new label, and then load, say, continue training on the previous network. So you've got a network that works well, it's not working so well for these new cells, mask up some of these new cells, and then do some additional training, maybe just an extra hour or so of the network processing these new images, and that will make it adapt to those neurons as well. And theoretically, this could lead to having something that's a little bit more robust across different neuron populations. So you don't once you've done this training, it's not like you're stuck there and you have to start again for a new data set. You could just build on this network. And then, this 5,000 iterations is a reasonable starting point. We just need to give it a name, and it'll save a screenshot. So this is if I click train now, it'll start to train the network. And we haven't really got time to get too much into this, but it's learning that from the image it's given what masks it should present. And so after, we've got the masks already given to it, the ground truth data. And so we can, over time, train the network and see how different its results are, and slowly converge to a point where it's creating masks that are very similar to the masks that we gave it. And this is why the image input data that you use later on when you're using a network, it should be the same type of image data, the same objective, same setting, not necessarily exposure times, but the same spatial settings, binning and objective, so that the network understands what it's seeing. I'm just going to finish it now, but you can see that we have it running on the GPU. And when we run these, we use actually a lot of times we'll have it maybe 8 to 12 hours is usually enough time to get a stable result. Okay, so that's how you would train the network. And we already sort of talked about how to use the network, inside a job. Just so you know, another way of using the general analysis, underneath image, this is where you can find the general analysis tools outside of jobs. So you may have a situation where you don't want to use jobs, you just want to analyze some data that you already have. So you can do that in here. This is where you can create a new jobs, a GA3 recipe. Let me show you from the Explorer. So if I go to the Analysis Explorer, here are the jobs that we have available on the microscope. And this is one that I made to sort of demonstrate analyzing these neurons. So if I click edit, we've got our same sort of CSV stuff over here, so just don't worry about that. What I'm doing is I've got the 555 channel. I have our segment AI that we looked at. And actually, I just want to point out some things for here so that you have a good chance of using this well. I'm using the one we already have trained for dealing with this data. And I'm just doing some smoothing and some cleaning and then separating. So if I turn this off, you can see that I have some issues here with cells that are joined together. If I just put a separate on, those cells are now separated. And if I wanted to, I could filter on size and circularity or things like that before I went deeper into the pipeline here. And then I'm just coloring by ID so that I can see those cells clearly. And then here it's just like if I wanted to filter based on something, I could start to filter. Here I'm filtering based on area, so I could start to get rid of cells if they were too big or too small and things like that. And here is a huge list of the sort of things you could filter on. Yeah, and then that would be it. So this is how you would build it in jobs. If you wanted to run it separately as a batch, then you would use the batch tool just here under image, batch GA3, and you would be able to select a folder. So in here, you could just add a folder to where we so I would say, okay, here's my AI. Here's my folder with all the images. And then I could run that, and that would analyze the images and create the CSV files. So kind of like the same way that you would run a macro or something with ImageJ. Yeah, that's all I really wanted to go through today. Did anyone have any questions about what we went through? Okay, thank you for coming.